Analysis of variance is a collection of statistical models used to analyze the differences among group means and their associated procedures. Developed by statistician and evolutionary biologist Ronald Fisher. In the ANOVA setting, the observed variance in a particular variable is partitioned into components attributable to different sources of variation. In its simplest form, ANOVA provides a statistical test of whether or not the means of several groups are equal, and therefore generalizes the t-test to more than two groups. ANOVAs are useful for comparing three or more means for statistical significance. It is conceptually similar to multiple two-sample t-tests, but is less conservative and is therefore suited to a wide range of practical problems. History. While the analysis of variance reached fruition in the 20th century, antecedents extend centuries into the past according to Stigler. These include hypothesis testing, the partitioning of sums of squares, experimental techniques and the additive model. Laplace was performing hypothesis testing in the 1770s. The development of least squares methods by Laplace and Gauss circa 1800 provided an improved method of combining observations. It also initiated much study of the contributions to sums of squares. Laplace soon knew how to estimate a variance from a residual sum of squares. By 1827, Laplace was using least squares methods to address ANOVA problems regarding measurements of atmospheric tides. Before 1800, astronomers had isolated observational errors resulting from reaction times and had developed methods of reducing the errors. The experimental methods used in the study of the personal equation were later accepted by the emerging field of psychology, which developed strong experimental methods to which randomization and blinding were soon added. An eloquent non-mathematical explanation of the additive effects model was available in 1885. Ronald Fisher introduced the term variance and proposed its formal analysis in a 1918 article, The Correlation Between Relatives on the Supposition of Mendelian Inheritance. His first application of the analysis of variance was published in 1921. Analysis of variance became widely known after being included in Fisher's 1925 book Statistical Methods for Research Workers. Randomization models were developed by several researchers. The first was published in Polish by Neyman in 1923. One of the attributes of ANOVA which ensured its early popularity was computational elegance. The structure of the additive model allows solution for the additive coefficients by simple algebra rather than by matrix calculations. In the era of mechanical calculators, this simplicity was critical. The determination of statistical significance also required access to tables of the f-function which were supplied by early statistics texts. Motivating example. The analysis of variance can be used as an exploratory tool to explain observations. A dog show provides an example. A dog show is not a random sampling of the breed. It is typically limited to dogs that are adult, purebred, and exemplary. A histogram of dog weights from a show might plausibly be rather complex, like the yellow-orange distribution shown in the illustrations. Suppose we wanted to predict the weight of a dog based on a certain set of characteristics of each dog. Before we could do that, we would need to explain the distribution of weights by dividing the dog population into groups based on those characteristics. A successful grouping will split dogs such that each group has a low variance of dog weights and the mean of each group is distinct. In the illustrations to the right, each group is identified as x1, x2, etc. In the first illustration, we divide the dogs according to the product of two binary groupings. Young versus old, and short-haired versus long-haired. Since the distributions of dog weight within each of the groups has a large variance, and since the means are very close across groups, grouping dogs by these characteristics does not produce an effective way to explain the variation in dog weights. Knowing which group a dog is in does not allow us to make any reasonable statements as to what that dog's weight is likely to be. Thus, this grouping fails to fit the distribution we are trying to explain. An attempt to explain the weight distribution by grouping dogs as in would probably be somewhat more successful. The heaviest show dogs are likely to be big, strong working breeds, while breeds kept as pets tend to be smaller and thus lighter. As shown by the second illustration, the distributions have variances that are considerably smaller than in the first case, and the means are more reasonably distinguishable. However, the significant overlap of distributions, for example, means that we cannot reliably say that x1 and x2 are truly distinct. An attempt to explain weight by breed is likely to produce a very good fit. All Chihuahuas are light and all St. Bernards are heavy. The difference in weight between setters and pointers does not justify separate breeds. The analysis of variance provides the formal tools to justify these intuitive judgments. A common use of the method is the analysis of experimental data or the development of models. The method has some advantages over correlation. Not all of the data must be numeric, and one result of the method is a judgment in the confidence in an explanatory relationship, background and terminology. ANOVA is a particular form of statistical hypothesis testing heavily used in the analysis of experimental data. A test result is called statistically significant if it is deemed unlikely to have occurred by chance, assuming the truth of the null hypothesis. A statistically significant result, when a probability is less than a threshold, justifies the rejection of the null hypothesis, but only if the a priori probability of the null hypothesis is not high. In the typical application of ANOVA, the null hypothesis is that all groups are simply random samples of the same population. For example, when studying the effect of different treatments on similar samples of patients, the null hypothesis would be that all treatments have the same effect. Rejecting the null hypothesis would imply that different treatments result in altered effects. By construction, hypothesis testing limits the rate of type 1 errors to a significance level. Experimenters also wish to limit type 2 errors. The rate of type 2 errors depends largely on sample size, significance level and effect size. The terminology of ANOVA is largely from the statistical design of experiments. The experimenter adjusts factors and measures responses in an attempt to determine an effect. Factors are assigned to experimental units by a combination of randomization and blocking to ensure the validity of the results. Blinding keeps the weighing impartial. Responses show a variability that is partially the result of the effect and is partially random error. ANOVA is the synthesis of several ideas and it is used for multiple purposes. As a consequence, it is difficult to define concisely or precisely. Classical ANOVA for balanced data does three things at once. As exploratory data analysis, an ANOVA is an organization of an additive data decomposition, and its sums of squares indicate the variance of each component of the decomposition. Comparisons of mean squares, along with f-tests, allow testing of a nested sequence of models. Closely related to the ANOVA is a linear model fit with coefficient estimates and standard errors. In short, ANOVA is a statistical tool used in several ways to develop and confirm an explanation for the observed data. Additionally, it is computationally elegant and relatively robust against violations of its assumptions. ANOVA provides industrial-strength statistical analysis. It has been adapted to the analysis of a variety of experimental designs. As a result, ANOVA has long enjoyed the status of being the most used statistical technique in psychological research. ANOVA is probably the most useful technique in the field of statistical inference. ANOVA is difficult to teach, particularly for complex experiments, with split plot designs being notorious. In some cases, the proper application of the method is best determined by problem pattern recognition followed by the consultation of a classic, authoritative test. Design of experiments terms balanced design and experimental design where all cells have the same number of observations. blocking a schedule for conducting treatment combinations in an experimental study such that any effects on the experimental results due to a known change in raw materials, operators, machines, etc., become concentrated in the levels of the blocking variable. The reason for blocking is to isolate a systematic effect and prevent it from obscuring the main effects. Blocking is achieved by restricting randomization. Design a set of experimental runs which allows the fit of a particular model and the estimate of effects. Design of experiments. An approach to problem solving involving collection of data that will support valid, defensible, and supportable conclusions. Effect: how changing the settings of the factor changes the response. The effect of a single factor is also called a main effect. Error: unexplained variation in a collection of observations. Those typically require understanding of both random error and lack of fit in error. Experimental unit: the entity to which a specific treatment combination is applied. Factors: process inputs an investigator manipulates to cause a change in the output. Lack of fit in error: error that occurs when the analysis omits one or more important terms or factors from the process model, including replication in a DO allows separation of experimental error into its components. Lack of fit and random error. Model: mathematical relationship which relates changes in a given response to changes in one or more factors. Random error: error that occurs due to natural variation in the process. Random error is typically assumed to be normally distributed with zero mean and a constant variance. Random error is also called experimental error. Randomization: a schedule for allocating treatment material and for conducting treatment combinations in a DO such that the conditions in one run neither depend on the conditions of the previous run nor predict the conditions in the subsequent runs. Replication: performing the same treatment combination more than once, including replication, allows an estimate of the random error independent of any lack of fit in error. Response is the output of a process, sometimes called dependent variable. Treatment: a treatment is a specific combination of factor levels whose effect is to be compared with other treatments. Classes of models. There are three classes of models used in the analysis of variance, and these are outlined here. Fixed effects models. The fixed effects model of analysis of variance applies to situations in which the experimenter applies one or more treatments to the subjects of the experiment to see whether the response variable values change. This allows the experimenter to estimate the ranges of response variable values that the treatment would generate in the population as a whole. Random effects models. Random effects model is used when the treatments are not fixed. This occurs when the various factor levels are sampled from a larger population. Because the levels themselves are random variables, some assumptions and the method of contrasting the treatments differ from the fixed effects model. Mixed effects models. A mixed effects model contains experimental factors of both fixed and random effects types, with appropriately different interpretations and analysis for the two types. Example: teaching experiments could be performed by a university department to find a good introductory textbook, with each text considered a treatment. The fixed effects model would compare a list of candidate texts. The random effects model would determine whether important differences exist among a list of randomly selected texts. The mixed effects model would compare the incumbent texts to randomly selected alternatives. Defining fixed and random effects has proven elusive, with competing definitions arguably leading toward a linguistic quagmire.